Okay, so students, today we are going to start with the new class of the anti-cancer agent that is called as the antimetabolites. So first we will see what antimetabolites are. In the previous lectures, we have covered one major class, that is the DNA alkylators, right? In DNA alkylating agents, we covered 4 to 5 different subclasses. That is nitrogen mustards, nitrosoureas, DNA methylating agent, and one more class we have covered. So likewise, we have covered the DNA alkylating agent. And now the next major class of these anti-cancer agents is the antimetabolites. So whatever the antineoplastic agent we discussed so far, they only damage the existing DNA, right? Whatever the DNA which is synthesized during the cell cycle. So whatever the antineoplastic agent we discussed so far, they damage the existing DNA and in the less concentration to the RNA, and thereby they inhibit the DNA replication process. The antimetabolites are the anti-cancer agent. They directly inhibit the DNA synthesis by the formation of the nucleotides. Okay, so the antineoplastic agent, which we had discussed by yesterday, they damage the existing DNA, whereas the antimetabolite anti-cancer agent, they inhibit the synthesis of DNA by the formation of the nucleotides. So these antimetabolites agents, they inhibit the synthesis of the DNA by the formation of the nucleotide. Now, how they inhibit the synthesis of the nucleotide? How they inhibit the synthesis of the DNA? That there is a enzyme. We will check out the name of the enzyme in the next slide. So these agents, they inhibit enzyme, and thereby they inhibit the synthesis of the DNA. So these antimetabolite agent, what they do, they bind to the active site of the enzyme. They bind to the active site of the enzyme and thereby inhibit the binding of the active substrate to that proper active site of the enzyme and thereby inhibit the DNA synthesis. Generally, these antimetabolite agents, they exist as a prodrug and they require bioactivation. And these antimetabolite agents, they are cell specific. Generally they act on the S phase of the cancer cell cycle. Now these antimetabolic antineoplastics, they are categorized into the different classes based on the nucleotide which they inhibit. Okay, based on the nucleotide which they inhibit. For example, purine antagonist. For example, purine antagonist. These agents inhibit the synthesis of purine-based nucleotides. Now, which one is a purine-based nucleotide? That is the AMP, that is adenosine monophosphate. Then the second one is the guanosine monophosphate, that is the GMP. So these are what? These are the purine-based nucleotides, right? So these anti-metabolite antineoplastic agents, they are categorized into 3 to 4 different classes based on the which nucleotide they inhibit. For example, the first one is the purine antagonist. Now this purine antagonist agent, they inhibit the synthesis of the purine based nucleotides, for example AMP and the GMP. Second one is the pyrimidine antagonist. So as the name suggests, these agent inhibit the production of the pyrimidine based nucleotides. For example, deoxythymidine monophosphate, that is a DTMP. So these are the basic nucleotides, that is a purine based and the pyrimidine based nucleotides. So these antimetabolite antineoplastic agents, they inhibit these nucleotides, that is the purine and the pyrimidine. So which are the purine antagonist? That is the AMP and the GMP. Which are the pyrimidine antagonist? That is the DTMP. Okay. So we are going to discuss this in brief. So the first one is the pyrimidine antimetabolite. That is, these antimetabolites, they inhibit these pyrimidine bases. These antimetabolite agents, they inhibit these pyrimidine bases. Okay, or the pyrimidine nucleotides. So the first one is the pyrimidine antagonist. So as we have discussed, pyrimidine antagonist means what? They inhibit the synthesis of the DTMP, that is the deoxythymidine monophosphate. So the name of the class is the pyrimidine antagonist or the DTMP synthesis inhibitors, that is the deoxythymidine monophosphate synthesis inhibitors. Okay. So before studying this in brief, first we will discuss how the DTMP biosynthesis takes place. So this is the DTMP synthesis pathway, that this DTMP, it is synthesized from the DUMP, that is the deoxyuridine monophosphate, which is present in front of you, right? The DTMP synthesis, it takes place from the DUMP, that is the deoxyuridine monophosphate. Now this DTMP, it is produced by C5 methylation of the DUMP. So this is the carbon number 5 of this pyrimidine base. If you start the numbering from this nitrogen, so it is 1, 2, 3, 4 and 5. So the DTMP synthesis, it takes place from the C5 methylation of the deoxyuridine monophosphate. If you check out both the structure of DUMP and the DTMP, so there is only one change, that is hydrogen is replaced by the methyl group. Otherwise, both the structures are same. But it is not such simple pathway, because in the replacement of hydrogen by methyl, it takes place various TD steps. So if you check out, so this DUMP, it is interact with the enzyme called as the thymidylate synthase, and there is a factor, and there is a factor. Now the name of this factor is 510 methylene tetrahydrofolate, that is a 510 THF. The name of the factor is 510 methylene tetrahydrofolate. So this factor, this enzyme and this substrate, so by collectively, by this process, the DTMP is get synthesized. So it's a simple process for the synthesis of DTMP from DUMP, that is the C5 methylation of the DUMP. The rate limiting enzyme of the DTMP synthetic pathway is the sulfhydryl containing thymidylate synthase. Sulfhydryl containing thymidylate synthase with 510 tetramethylene 510 tetramethylene 510 methylene tetrahydrofolate. The name of the factor is 510 methylene tetrahydrofolate, which serving as a methyl donating, this factor it serve as a methyl donating cofactor. Now all the DTMP synthesis inhibitors, that is our agents, the most common pyrimidine antagonist agent is the 5-fluorouracil. Okay, so uh, uh, all these DTMP synthesis inhibitors, they inhibit this thymidylate synthase either directly or the indirectly, and this will result in the thymineless death in the actively dividing cell. So without DTMP, without the synthesis of DMP, and its deoxythymidine triphosphate metabolite, DNA will fragment and the cell will die. Because for the survival of the cell, this DTMP is required. So if this DTMP synthesis is get inhibited, then the cell will die. So generally, our these DTMP inhibitors, they generally act on this enzyme, that is the thymidylate synthase enzyme, and thereby inhibit the synthesis of the DTMP. Now we will briefly check out this pathway. Now this synthase enzyme, this thymidylate synthase enzyme, it is very large. This enzyme is very large, and it contains a deep pocket for the binding of the substrate as well as the cofactor. Okay. For example, if we take an example of a cooking pot, for example, cooking pot. So in that cooking pot, we added the ingredients like the cofactor and the substrate, and the process of making product is begin. So what is the product? That is the DTMP, right? So this thymidylate synthase enzyme, it's very large, and it is having very big pockets for the binding of the substrate and for the binding of the cofactor. The active site binding motifs for both the substrate and the cofactor. So it can easily accommodate that enzyme, thymidylate synthase enzyme. It can easily accommodate your cofactor as well as the substrate. So there are various interaction takes place when the substrate and the enzyme bind with each other. So these are the various interactions. So it is shown in the figure. So if you check out this aspargine or the aspartic acid 226, it forms essential hydrogen bond with the DUMP pyrimidine 4-oxo, right? This aspargine 226, it forms a hydrogen bond with the 4-oxo group of this DUMP substrate, and also it forms hydrogen bond with the N3H group. This is the third nitrogen, no? And hence N3H group. So this aspargine 226, it forms hydrogen bond with this carboxy group and the N3 hydrogen group of this DUMP substrate. Whereas histidine 196, it also forms a hydrogen bond with the pyrimidine 4-oxo moieties. So this is our substrate, right? DUMP is our substrate which bind to the thymidylate synthase enzyme. So whatever these amino acids which I have mentioned, all the amino acids are present in the active site of the thymidylate synthase enzyme, right? So histidine 196, it also form a hydrogen bond with the pyrimidine 4-oxo moiety. Then the main chain amide of aspartic acid 218, this one, aspartic acid 218, it form a hydrogen bond with the pyrimidine 2-oxo moiety of this DUMP. Then the four arginine residues like the arginine 215, arginine 176, arginine 175, and arginine 50, they form the electrostatic bond with the anionic deoxyribose 5-phosphate. So all these arginine residues, they form an electrostatic bond with these anionic residues of the phosphate. And tyrosine and the histidine. Now this one, tyrosine and the histidine, these residues, they form hydrogen bond with the 3 hydroxy group of this sugar. right? Because the numbering start from this oxygen. So 1, 2, 3. So they form bond with these 3 hydroxy sugar, right? So this is how the interaction between DUMP and the enzyme takes place. Similarly, over here, the interaction between the 510 methylene THF, that is our cofactor 510 methylene tetrahydrofolate, and the enzyme they have mentioned over here, and thus this ternary complex is formed. Thus, this ternary complex is formed by forming interaction between DUMP, thymidylate synthase, and the cofactor. Right? So, how this pathway takes place? So, the C6 position, the carbon number 6 position of this DUMP substrate, it is surrounded by electron withdrawing nitrogen and the oxygen atoms. So because of this carbon become highly electrophilic. It becomes electron deficient because this nitrogen and oxygen atom, they pull up the electrons from this carbon. And as this carbon, the carbon number 6 of this DUMP substrate, it has become highly electrophilic. Once it becomes highly electrophilic, then it can easily attack by the nucleophilic cysteine 195 of the thymidylate synthase. The cysteine sulfhydryl group, it launches an intermolecular nucleophilic attack, forming a new covalent bond. Check it out over here. So they form a new covalent bond. So this is the, if you check out this pyrimidine ring, and to this pyrimidine ring, the sulfur group is get attached. As the carbon number 6 is become electrophilic, so the nucleophilic group can easily attack on this electrophilic group, and thereby they form a bond. Okay, they form a bond, a covalent bond. Now the bond that break, that is which bond is breaked when this sulfhydryl group is get attached to this 6th position. So which bond is break? That is the double bond between carbon number 5 and 6. It is get breaked. And this bond, the electrons of this bond attacks the methylene group of the cofactor. Okay, so if you check out, the bond breaks and the electrons of this bond, they attack on the methylene group. So this is the methylene group of the cofactor, with the release of the N10 nitrogen. So the bond, when the electrons of this 5,6 double bond, when the bond breaks, so the electrons, they attack on this methylene group of the cofactor. While attacking, they release the N10, this nitrogen 10 group, and the cofactor imidazoline ring, it is get break. So the ring get open. So taken together, all these steps, that is the step number 1, step number 2, and this step number 3. Which is the step number 3? That is the breaking of the bond. So when we take these all the steps together, a ternary complex of enzyme, substrate, and the cofactor, it is get synthesized. Okay, this ternary complexes get synthesized. So check out properly how the ternary complex is formed. Now, a series of reactions involving, now there are various reaction takes place which involve bond breaking and bond making. It is shown over here in the ternary complex. Finally, it leads to the formation of the DTMP 78 DHF and a regenerated thymidylate synthase. So this thymidylate synthase enzyme, it is again regenerated. Once this thymidylate synthase enzyme is regenerated, then again it acts on another DUMP substrate to form the ternary complex. So this is all these steps are the reversible reactions. Okay. So various bond breaking and bond breaking steps takes place to form the DTMP, 7,8-DHF and the regenerated thymidylate synthase. Now in this step, the C5H abstraction. Now this hydrogen is present in the structure of the DUMP, which is also present in the structure of the ternary complex. Right? Now this hydrogen which is present at carbon number 5, it is abstracted by the N10 of the cofactor. Okay. Now this N10 which is released, right? This N10 is get released. So it is shown over here to open the ring. So this this hydrogen is get abstracted by this N10 nitrogen. N10 nitrogen, and this step is essential for the synthesis of the DTMP. This hydrogen abstraction step, it is essential for the synthesis of the DTMP. To complete the biochemical cycle, 7,8-DHF, the 7,8-DHF, it is must reduced to the THF, that is the tetrahydrofolate, via DHFR enzyme using NADPH. And finally, this THF is get converted to 5,10 methylene tetrahydrofolate, which is our factor. So this THF, it is get again converted into our cofactor, that is the 5,10 THF, through the action of SH-empty pyridoxal pyrophosphate. SH-empty means what? That is a serine hydroxy methyl transferase. Serine hydroxy methyl transferase, and with the help of vitamin B6, this THF, it is get again converted in our cofactor, that is 5,10 methylene THF. So this is how the biosynthesis of DTMP takes place from the DUMP. Fine. Now the first agent of the pyrimidine analog is the fluorouracil. Now if you check out over here, it is shown the activation of the fluorouracil. If you check out properly, then this fluorouracil is it is a prodrug. Now then that fluorouracil prodrug, it is get converted into its the active form. So to bind to the thymidylate synthase enzyme, this fluorinated pyrimidine prodrug, it is get converted into its deoxyribonucleotide form. So this is its deoxyribonucleotide form. So the active form of the fluorouracil, it differ from the endogenous substrate only by the presence of the 5-fluoro group. So this is how the fluorouracil prodrug is get converted into its deoxyuridine monophosphate group, which is the active group of the fluorouracil. So the first step is the fluorouracil is get converted into the 5-fluorouridine monophosphate, 5-fluorouridine monophosphate by the enzyme oroate phosphoribosyl transferase. So phosphoribosyl transferase. So this group add the ribose sugar and the phosphate group to this fluorouracil prodrug to form this 5-fluorouridine monophosphate. Then by the enzyme uridine monophosphate kinase, by the enzyme uridine monophosphate kinase, it add one more phosphate group over here to form 5-fluoro 5-fluorouridine diphosphate. This is what? Monophosphate. This is what? Diphosphate. Then by the enzyme ribonucleotide reductase, by the enzyme ribonucleotide reductase, it get synthesized into the 5-fluorodeoxyuridine diphosphate. Now this reductase enzyme, it remove one hydroxy group from this sugar. Okay, it remove one hydroxy group from this sugar to form this intermediate product. Then by the phosphatase enzyme, it gets synthesized our active drug of the fluorouracil, that's the 5-fluorodeoxyuridine monophosphate. So which is the active drug of the 5-fluorouracil. In this step, whatever the new phosphate group which is added by the UMP kinase, it is get removed to form our active drug, that is the 5-fluorodeoxyuridine monophosphate. Then later on, this is the mechanism of action of the fluorouracil, which is shown again in this figure. So again, this pathway is little bit blur. So you have to check out this pathway in the FOA book properly. So as discussed, the structures of the 5-fluoro-DUMP and the structure of these DUMP, which we check out in that pathway, both the structures are similar. Only the change is what? In that substrate, there was a hydrogen group, and in this 5-fluoro-DUMP, there is a fluorine group. Okay. So as discussed in earlier slides, what happened, that is this substrate interact with the thymidylate synthase enzyme and with the cofactor to form a ternary complex. Once ternary complex is get generated, so from that ternary complex, our the 7,8 DHF is get synthesized. But there is a mandatory step, or which is the mandatory step? That is the abstraction of a hydrogen which is present at the carbon number 5 of this substrate. It should be abstracted by the cofactor. But now in this structure of 5-fluoro-DUMP, there is no any hydrogen, there is only fluorine. So it does not abstract the fluorine because it want hydrogen to form the 7,8-DHF. But in this case, there is a fluorine at the fifth position. So the abstraction of hydrogen does not take place. So the synthesis of 7,8-DHF is not takes place, and hence the cell will die because the 7,8-DHF then THF, it is not get synthesized. And for the survival of the cell or for the synthesis of DNA, that 7,8-DHF is required. So the 7,8-DHF is not synthesized, so the cell will die.